The sliding filament theory is one of the most important theories that ties together an important idea behind why and how our muscles and bodies work. The sliding filament theory was created from a large list of scientists all from different time periods and their individual research done on structures of muscles, the microbiology within the human body, and a variety of other fields. However, formally speaking, the actual sliding filament theory was formulated by Hugh Huxley and John Hansen, both biophysicists. Their theory can be summarized with a subsequent model, which models what muscle contraction looks like on a more micro level and how force is developed and used in this process. However, before explaining the sliding filament theory, it is important to understand some of the basic anatomy structure of muscles and their physiology. Muscles can be broken down into fascicles, which is another term for bundles of muscle fibers, then myofibrils, then sarcomeres, descending in order from largest to smallest. In this video, myofibrils and sarcomeres are essentially the depth in which the sliding filament theory lies. Focusing in on myofibrils, this is essentially the layer of muscle structure in which the sliding filament theory applies. In each muscle fiber is a combination of many myofibrils, both thick and thin. The thin filaments being formed with the protein actin and the thick filaments being formed with the protein myosin. This combination of both types of myofibrils is what causes the familiar striations found in muscles. However, it is the movement of these two types of myofibrils that is actually important and lies as the foundation of the sliding filament theory. To put the theory into simpler terms, it is essentially a theory that relies on the idea that tension is generated through the movement of myofibrils with thin filaments slide over thick filaments, shortening the entire myofibril. This movement takes place specifically in the sarcomere. While this reaction may seem simple in definition, there are many steps to this in actuality. Beginning with a resting sarcomere, the active site is exposed, then a myosin head attaches to create a cross bridge, which then pivots to cause a sliding until the reaction stops and the myosin head detaches. The process of sliding filaments first begins with a motor neuron stimulating a muscle fiber. The signal from the neuron travels through the axon and to the neuromuscular junction, which connect the neuron to the muscles. This signal depolarizes the T-tubular membrane of the cell, which then allows the calcium ions stored within the sarcoplasmic reticulum to be released into actin filaments. Looking closer at it, this is caused by an interaction between the dihydropyridine and ryanodine receptors, which is then facilitated by the STAC3 protein. Moving on to the next step, the calcium ions previously mentioned that have now been released into the space of filaments is now traveling through the space between. The calcium binds eventually to the troponin C, which then causes the tropomyosin cable portion to move away from the active sites on actin filaments, allowing myosin heads to attach or bind, forming the titular cross bridges. After the cross bridges are formed, the release of ATP ions allows for the myosin heads to pivot, bending and pulling the actin filaments with it. This causes the overlap of actin over myosin and the contraction of the skeletal muscle. During the contraction, many byproducts are